Alright guys, what this next video series is going to deal with the cell cycle, and we're in biology. So this is going to cover the basics of the cell cycle. So let's get started. Alright, so just what is this cell cycle? Well, the cell cycle is going to cover from the birth of the cell, okay, so this is a generic cell that I have here in this little picture, okay, all the way through the whole entire process of when this cell goes through division, okay? And during the cell cycle, when this cell divides, we're going to have two identical cells that are going to be formed, is what's going to happen. So the cell cycle is going to go from the minute that this cell is born all the way through the whole entire life cycle of the cell until it actually replicates itself. And we're going to get out of the cell cycle through the process called mitosis. We're going to have an exact copy of that same cell that we were originally dealing with. So if this is a skin cell, okay, so this is a skin cell, all right, this would be the parent skin cell. I'm going to form two exact daughter cells. So this is going to be a daughter cell. This is going to be a daughter cell. But these are exact copies of that same skin cell that we had before. All right, so this would be like a little keratinocyte or something like that that's hanging out in our skin. So that means it's going to have the exact same number of chromosomes. Remember, we're humans, so how many chromosomes do we have? We have 46 chromosomes. So in here, I'm going to have 46 chromosomes, and these two daughter cells are both going to have 46 chromosomes. Cells are going to undergo this process, okay, that we're talking about when we talk about the cell cycle in mitosis. They're going to go through this process. Why would a cell go through the cell cycle? Well, at points in your life, in an organism's life, it's going to go through the cell cycle in order to grow and to repair itself. All right? So this process is going to be for growth, okay, in order to grow and to repair itself. All right? When I talk about cell growth, all right, we often think of growth as just like increasing in size. So I have a cell that's like this, and this cell eventually grew and it became bigger like this. So it just became a bigger cell. But another way to think about cell growth is we have a cell that's like this, and then this cell is going to increase in number. Okay, so now we have a cell that looks like this, and we have a cell that looks like this. And then these cells divide, okay, into two more cells that are both exact copies of the previous cell. All right, so another way to think about growth, all right, when you grow in size, okay, as a human being, you're increasing in cell number. You have to think about this particular process, all right, initially sperm met egg. Okay, and this would have been you. This would have been you way back in the day when sperm met egg. And then what happened is this cell began to divide, okay, and then these cells began to divide, and eventually wound up with a trillion cells that's going to make you. Now, this becomes a very complex process in that some of these cells begin to differentiate into your nervous system, your skeletal system, your cardiovascular system, and etc. But the same concept is you grew. You started out as one cell, and then you became two cells, and then those two cells became four cells, and then it eventually make our way to a trillion different cells that we have. So the point of going through the cell cycle in mitosis is for growth and repair. The repair component of it is if tissue gets injured, okay, or tissue gets destroyed, and we want to grow this particular tissue back to replace itself, okay, to start anew is what we can do with this tissue repair when we take a look at it. So this is the point of the cell cycle in mitosis, is for growth and repair. And in this process, we're going to create an identical copy of that previous cell. So let's take a look at the cell cycle. The cell cycle, we're going to make a little circle. It's going to look something like this. It's going to go just like this right here. Okay. And the first stage of the cell cycle is called interphase. So this is called interphase. I have to spread this out. So this is all interphase that I have right here. This is interphase that's going on right here. And interphase consists of the G1 stage, okay? It consists of the S stage, and it consists of the G2 stage, okay? These are the major stages that I have for interphase, all right? So this is all going to be interphase. It's going to be right here, all right? Let me try to draw this a little bit better. But this is interphase that we have going on right here. The cell spends about 90%, okay, of its time in interphase. All right? And then we have this other little phase that's way down here that we're going to call the M phase. Okay? So this phase right here is going to be called the M phase. And the M phase is going to deal with the process called mitosis. And mitosis has some stages of its own that we call prophase, that we call metaphase, that we call anaphase, and we call telophase. If you can remember PMAT, okay, PMAT, we can kind of remember that. But we're going to focus in on interphase right now is what we're going to focus in on. So we have this cell that's just been born. So we have a brand new cell that's just going to be born. And anytime that we're in interphase, we can see the nucleus. So I know that I'm in interphase as far as the cell is concerned when I can see the nucleus. All right? And I'm not able to see chromosomes present in that particular cell yet. All right? But there are some things that are happening inside this nucleus as this cell is going through the G1 stage. So during the G1 stage, all right, G1, the cell is going to grow. Okay? So G1 of interphase, the cell is going to grow. And these structures that are called centrioles are going to replicate. All right, so G1, basically the cell is going to grow. Okay, we talk about growth, we're talking about increasing in size, we're not increasing cell number in the G1 phase. And these structures are called centrioles are going to replicate themselves. That's what's going to happen in the G1 stage. This is what inside of a cell, so I'll kind of draw this right here. So this is my cell right here. Here's the nucleus, okay? And we have these structures that are called centrioles, okay? There's little structures down here. They're going to replicate, is what they're going to do. A centriole is going to come in and depart in just a little bit more because these structures are going to help move chromosomes around is what they're going to do. But that's what's happening in the G1 stage. So the G1 stage, the cell is growing. These things called centrioles are going to replicate is what they're going to do. Now this G1 stage can take a long time. Sometimes biologists also include within the G1 stage, they include something called the Geo stage. Okay? So inside here, sometimes you'll see something also written called the Geo stage, which a cell kind of goes into and kind of rests in this Geo stage before it moves on to the S phase. All right. Once we get to the S phase, what's going to happen in the S phase? S stands for synthesis. So when I synthesize something, that means that I'm going to make something. You have to understand that inside of your nucleus, you have DNA. Okay, that's kind of hanging out in the form of what we call chromatin. And I'm going to show you what chromatin looks like here just right now. So I'm going to draw a giant cell is what I'm going to draw. Just a giant, typical animal cell that they say, which really doesn't exist, a typical animal cell. But okay, so this is my animal cell that I have right here. And then right here is my nucleus that I have right here. All right, inside the nucleus, all right, during interphase, the DNA is just kind of hanging out is what it's kind of doing. Okay, it's kind of loosely structured is what it's going to do. All right, so if I can have, let me get a little smaller piece here of thread. So this will be my DNA that's right here. So this is my DNA. And you can see this DNA as it's hanging out inside the cell. It's real loosely, okay? It's real loose and it's not like tightly wound together, all right? What this is what we call chromatin. And what chromatin is, okay, so this is chromatin. It's loosely wound DNA is basically what it is. It's loosely wound DNA that's wrapped around proteins and things like that. But it's during this S phase, okay, so during this S phase is when this DNA is going to replicate itself. This DNA is going to replicate itself, all right? So we have DNA that looks like this. It replicates itself. It copies itself. So now we have the exact same copy of this DNA as well. So now we have two copies of this exact same DNA. Now you have to understand as we go through this process, eventually we're going to start talking about chromosomes. And what happens to this chromatin is this chromatin starts to condense on itself and starts to collapse on itself and starts to become a lot thicker so you can actually see it. And then we wound up with a structure that's called a chromosome. We wind up with a structure that's called a chromosome. And here I'm going to represent a chromosome that kind of looks just like that. But during this stage, okay, during this stage of the S phase, we have copied this DNA. We have copied this DNA, and because we've copied this DNA, we have 2 exact copies of this DNA, and eventually this DNA is going to condense on itself a little bit later, and we get into a phase called the M phase, but this DNA is basically going to replicate itself into structures, okay? Once it replicates itself, the DNA replicates itself, and we move on to the M phase. This DNA condenses into structures that we call chromosomes, okay? Into structures that we have called chromosomes. This, what I have right here, is a replicated chromosome. This is a replicated chromosome that I'm showing here. And what I mean by a replicated chromosome is, okay, you have a center structure here of a replicated chromosome that's called the centromere. So right here's the centromere. All right, this is a replicated chromosome. And these little end structures here are called chromatids. These little end structures here are called chromatids that we have right here. Okay, so that's what I have right here, is a replicated chromosome that's inside of the nucleus. But really during this S phase, it's not going to look like this. All right, it's going to be completely unwound like this. But slowly but surely what begins to happen is this begins to condense on itself and form chromosomes. But that's not till a little bit later stage in the cell cycle that we call the M phase, that we're going to talk about just a little bit later. So we're still in interphase now. We've gone through the G1 stage. We've gone through the S phase, okay, which is synthesis. Now we're going to move on to the G2 stage. During the G2 stage, remember, this is all interphase. And remember, during interphase, we can see the nucleus is present inside the cell. During the G2 phase, this is a checkpoint stage. And we're just checking inside this particular type of cell that we're looking at. We're making sure that everything is ready to go for division. Okay, so we're making sure during this stage that, hey, we've doubled the amount of chromosomes. We doubled the amount of DNA that's present in that cell. Okay, and everything else is ready to go. If the cell's not ready to go through division, okay, there's something wrong within the cell will actually destruct itself if something's not correct. So you'll go through cellular death, okay, if something's wrong in the G2 stage, at this checkpoint stage, okay. For whatever reason, a cancer cell can beat this G2 stage even though there's clearly something wrong within the cell that we'll talk a little bit later. So the G2 stage is going to be called basically a checkpoint stage to make sure that everything's ready for division. Now we're going to move into the M phase. And the M phase is where the cell is going to start to divide during the M phase. Okay? And the M phase is short for mitosis. Mitosis is the cellular division that we're talking about. And remember, we're going to remember the term P-M-A-T for PMAT. Okay? So it's going to go through prophase, metaphase, anaphase, telophase. If we remember, a cell is only going to be in the M phase for 10% of the time. The other 90% we're going to be in interphase. So let's take a look at prophase. So let's take a look at a cell in prophase is what we're going to look at now. So during prophase, all right, this is my cell that's right here. During prophase, the nucleus begins to disappear. Okay, so the nucleus is going to be able to disappear inside that cell. And then what you're going to be able to see is you're going to be able to see now chromosomes. And you're going to see replicated chromosomes. All right, so this organism that we're taking a look at is going to have 2 chromosomes. All right, chromosome here, and this is a second chromosome right here that we'll do in black that we have. So during prophase, all right, the nucleus disappears. Okay, the nucleus disappears and the chromosomes become visible. The chromosomes are now visible. So chromosomes visible. And I'm going to put out here, I'm going to put these little structures that we call the centrioles. Okay, remember these centrioles replicated themselves during the G1 stage. I'll put them out here. So that's prophase. prophase that we have there. The next stage that we're going to have is going to be called metaphase. So after prophase, we're going to move into metaphase. And what's going to happen is these chromosomes are going to be able to begin to line at the middle. Now, how they're able to align at the middle is you have these centrioles, okay, that have these spindle fibers that are attached to them. And these spindle fibers are kind of like fishing line. It's basically cytoskeleton material that you have. And these centrioles are these spindle fibers are going to attach to the centrioles, which is that middle part of the chromosome that's there. Okay? So these spindle fibers are going to attach to not the centrioles, the centromeres of the chromosome is what they're going to attach to. So that's my one chromosome there. This is my other chromosome I'm going to do in red right here. Remember, these are replicated chromosomes. So when I take a look at metaphase, we're going to meet in the middle. So metaphase, the chromosomes are in the middle. So these chromosomes have met in the middle is what's happening in metaphase. After metaphase, now I'm going to go through anaphase. And what's going to happen in anaphase is these chromosomes are going to be able to get pulled apart. So now I'm going to move on to anaphase. So we went from prophase to metaphase. Now I'm going to go into anaphase. And during anaphase, what's going to happen is these chromosomes are going to get pulled apart. So here's my centrioles, okay? And this is going to be the pulling apart right here, guys. All right, so I'm going to have this pulling apart right here. And I'm going to have this pulling apart right here. Let me draw it this way. So this chromosome is getting pulled apart. Same thing's happening right here. This chromosome is going to get pulled apart. This chromosome is going to get pulled apart. So these chromatids are now, this replicated chromosome has now been separated, is what we've done. So inside this cell, okay, basically what we've done is we pulled apart this replicated chromosome, okay? So this is a chromosome here, this is a chromosome here, this is a chromosome here, this is now officially a chromosome here. So right now in this cell, I have 4 chromosomes that are sitting right here, okay? Inside this cell, I have 2 chromosomes, but they're replicated chromosomes. These chromosomes are not replicated. So anaphase, all right, the chromosomes pull apart. Replicated chromosomes pull apart. Is what they're doing. So replicated chromosomes are going to pull apart. I often say anaphase, think Anakin Skywalker. Anakin Skywalker was from the Star Wars movie. He got torn between good and evil. So these chromosomes are getting torn apart, and they're going to go their separate ways. So now we're going to move into the final stage. It's called telophase. In telophase, think 2. We're starting to form 2 cells. So in telophase, we're now going to start to form 2 cells that we have right here. So telophase, we're going to start to form 2 cells is what we're going to do. The nucleus begins to reappear. Okay, and then this is where we're going to start to form these 2 cells. So I'm going to have my chromosome here, my chromosome here, chromosome here, my chromosome here. All right, and this is what I call telophase. Now eventually in telophase, what's going to happen is this cytoplasm that's in the cell is going to completely, okay, separate itself, and then we're going to eventually wind up with 2 complete cells is what we're going to have. So we're going to have 2 complete cells. Each cell has 2 chromosomes, okay, so this cell has 2 chromosomes, this cell has 2 chromosomes. Note they're not replicated chromosomes, but they are 2 chromosomes, all right? This is going to be an exact copy of this original cell that we had. When they finally split, we call that stage cytokinesis. They don't really classify cytokinesis as part of the M stage, but that's when you officially have those 2 separated cells, and this nucleus begins to fully form. Right there is what's going to happen. So those are the basic stages of mitosis. If you can remember PMAT, prophase, metaphase, anaphase, and telophase, then you're going to understand the actual process of mitosis. Remember, mitosis is occurring for cell growth, okay, or for organism growth and repair. Okay, organism growth and repair is when this is actually going to occur. All right, what we're taking a look at here, guys, is this is an onion root tip. Okay, this is the root of an onion, and these are the various stages in this onion. of the cell cycle itself. So there's various stages of mitosis in here, as well as interphase is present here on these various slides that we have. So let's see if we can identify some of these different stages. First, let's identify some of these that are involved in interphase. Okay, so let's see if we can find some interphase ones. Remember, with interphase, what I'm trying to look for is with interphase, I'm trying to look for that I can definitely see the nucleus is present. So right here is going to be an interphase, and I'll put an I here for interphase. There's an interphase. There's an interphase here. Okay, that's an interphase. This is an interphase. All right, the majority of cells are going to be an interphase. There's interphase here. The nucleus is present when I'm taking a look at the cells. The next stage that I'm going to look for is going to be the first stage of mitosis that is called prophase. So right here is going to be a prophase. Okay, because prophase is when I'm able to see the chromosomes, but they're really not aligned in the middle yet. Right here's an early stage of prophase. It's right here. The chromosomes are becoming visible. Right here's an early stage of prophase. It's right here. This one right here is going to represent metaphase. Metaphase because the chromosomes are aligned at the middle. And then anaphase. Right here is probably the best representation. Right here is anaphase. So anaphase is torn between good and evil. These chromosomes are getting pulled apart. And then telophase. Let's see if we can find a telophase. Right here's another metaphase. There's a metaphase. Let's see if we can find a telophase. A good representation of telophase in which we have two cells that have formed. And probably right here is going to be a good representation of telophase between these two cells right here that I have is going to be a good representation of telophase. So those are the various stages that you can see of mitosis and interphase that we're able to see. Hopefully on your test you're able to do that and identify the various stages by looking at the diagrams. That's going to be one of the questions that I'm going to ask for you. So there is one thing that begins to happen that we kind of have to talk about. It's the second leading cause of death in the United States, and it's cancer. Cancer is basically a disease of uncontrolled cell division. So normally I have a cell. Okay, so normally if I have a cell here growing on this little tray right here, this cell is going to grow. It's going to divide. It's going to divide. It's going to divide until it reaches a certain population density, until it covers this little area right here. And we call that contact inhibition. So the cell immediately stops dividing once it reaches a certain population size or it covers that particular area. A cancer cell loses contact inhibition. So if this is a cancerous cell that's right here, it's going to continue to divide and it doesn't stop dividing. Okay, it divides, 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 it divides. It doesn't matter the size of the populations of cells that are in the surrounding area. It's just going to continue to divide. And when it divides, it can surround other tissue as well. And it can take the nutrients from them, and it can form what's called a tumor. The problem with a cancer cell is a cancer cell does not have a function. So as these cells divide and they overcrowd other tissues or other cells, that organ or that structure is going to lose its function itself. And if you have organs inside your body that lose their function, you have a chance of dying as a result of that. For whatever reason, a cancer cell, we take a look at the G2 stage of the cell cycle. Remember in the G2 stage, we took a look at it. If there is something wrong with the cell, it goes through apoptosis, and apoptosis is cellular death. For whatever reason, a cancer cell is able to continue to divide, and it goes through that G2 stage. Even though there's something wrong with the cell continues to divide, continues to divide, continues to divide. It doesn't undergo cellular death, even though something is wrong with that particular cell itself. So how does one get cancer? And specifically, most cancers arise as cells acquire a series of mutations, which are changes in this DNA that are related to uncontrolled cell division and causes them to divide a lot more quickly. So that's the basis, guys, of the cell cycle and mitosis that we took a look at. Remember, we go through this process, okay, for cell growth and repair. If you have any questions, don't hesitate to come see me in class. I'll be glad to answer them for you. Good luck in studying.